Okay, hello everyone, good evening. Most of the people are sleeping. Okay, good evening everyone. So friends, we are left with a little concept. Hello everyone. Friends, we are left with a very little concept of this particular sensory photobiology. We will finish this particular little part and we will move on to the solving questions. Okay, so I want you all people to be very patient. So it's going to be a long section. Okay, I want to be everyone to stay till the till the end of the session. Please be patient. Okay, that's great. Good evening everyone. Okay, let's continue. Okay, in the evening class we were discussing about the phototropism. If you all quickly recall, we understand the structure of cryptochrome, the structure of the phytochromes, and the structure of the phototropins. And if you all quickly recap, we discussed about how does this particular phototropins are involved in the movement, chloroplast movement. We understand the stomatal movement also. So friends, let's finish very quickly the last part of photoperiodism. I'll finish it in 15 minutes. Then we will move on to the solving all the PYQs. Okay, let's start. So I told you, right, this is the only area where we can expect the experimental based questions. Okay, so pay attention. So give me 15 minutes of time. First let me finish the entire experimental part. Once you have any kind of a doubt, I will solve each and every doubt. So pay attention. Good evening everyone. So photo period, how can you define the word photo period? Photo light, period time, or you can say that what is the relative length of the daylight and what is the relative length of the night. So friends, everyone listen to me carefully. How much amount of the daylight is required and how much amount of the night length is required for the plant to undergo the process of flowering. Now you can ask me a question, sir, does the light is really required for the process of flowering? Of course, yes. Not only flowering, friends, even for the dormancy conditions also. What do we require? Compulsory, we require this particular relative length of daylight and night. Let me explain you briefly. Okay, see, in the year 1920s, Garner and Allard, so friends listen to me 15 minutes, you will understand very clearly. Garner and Allard, they observed a large tobacco plant. Friends, originally what tobacco plants will be, all the students please keep it in your mind. Most of the tobacco plants, friends, they are very short. They are very short and they did not flower in the summer. I'm so sorry. Most of the tobacco plants will flower in the summer. Once again, keep it in your mind. Most of the tobacco plants are very short and friends, they are going to be flowered in the summer. But Garner and Allard observed that they have observed a very large tobacco plant. Originally all the tobacco plants are very short, friends, but they observed a very large tobacco plant that did not flower in the summer. Then see here, most tobacco plants flower in the summer, but whatever the tobacco plant that they observed, it is not flowering in the summer. You know, Garner and Allard, they have given the name for that particular tobacco plant as a Maryland mammoth. You know friends, what they understood is, first what they have done, all the students listen to me, they have carried out controlled experiments. What do you mean by the word controlled experiment? Suppose this is a plant, imagine this is the plant. This plant they packed in a containers. They packed in a containers. They manipulated. What they manipulated? Amount of the daylight and amount of the night also. So we can't understand, we can't plant cannot understand which is the daylight, which is the dark condition. So what they have done, they carried out the control experiments in the light boxes where they manipulated the amount of the light and dark. Now finally friends, they have understood if the plant wants to flower, every student listen my words carefully. If the plant wants to flower, minimum they require the day length 14 hours or less. 14 hours or less is minimum amount of the daylight that is required for the plant to flower. So that is what exactly we can call the critical day length. Everyone try to understand the definition of the critical day length. Critical day length means how much amount of the maximum light is required or how much amount of the minimum light is required for the plant to initiate the process of flowering. In general, an average, every plant require 12 to 14 hours of light or day length to initiate flowering. Suppose an example, any flower starts flowering by using the day length less than 12 to 14 hours, less than, then I can call it that particular plant as the short day plants. Any plants start to flower, if they want to flower, if they are using the day length more than 12 to 14 hours, more than 12 to 14 hours, what do we call it them as friends? We will be calling them as the long day plants. Then friends, what do you mean by the word day neutral plants? It doesn't matter whatever the day length it may be, it doesn't matter whatever the night length it may be. So they can easily flower, which means here you have to remember the light is not influencing the flowering in the day neutral plants. All the students please remember the examples without fail. All the students understand. When I can call a plant is the short day plant. Friends, if you understand these two paragraphs, then you can easily understand the experiments right now whatever we are going to discuss. Short day plants, as I said, plant that flower only when the day length is shorter than the critical day length. What is the critical day length? 12 to 14 hours. If the day length is less than this, obviously they can flower. So what's the best example? Chrysanthemum, some soya beans. Actually short day plants means what friends? They flower during the late summer or early winter. You know no friends, in the early winter what will happen? The day will be shorter and night length is getting increased. Next one, long day plants. Everyone listen. Plants that flower only when day length is longer than the critical day length, which means that when the plant is using more than 12 to 14 hours to initiate the process of flowering, then only we can call friends long day plants. Kindly don't forget example radish, lettuce and sorrel. Now friends, what is the unique characteristic here? Day neutral plant. What is the speciality of the day neutral plant? It is flowering is not at all influenced by the photoperiod, which means if the critical day length is more than the 12 to 14 hours or if the critical day length is less than the 12 to 14 hours, it doesn't matter. They are not going to get flowered at all. So flowering only occurs when they reach a particular stage of the maturity or certain external cue like temperature and water. When the temperature, water, once they reach the particular stage of maturity, they automatically undergo the process of flowering. It doesn't matter whether the critical day length is more than the normal, whether the critical day length is less than the normal, it normally undergo the flowering. Example rice as well as tomatoes. So friends, now I want every student has to understand from this particular slide. 1, 2, 3, 4. These 4 slides are very important where we can expect the experimental based questions. Just have a patience. I will explain in a very simple way. Listen to me carefully. I will explain every point twice. So pay attention. After that I'll give the time, I'll solve your doubt. Short day plant. Short day plant or long night plants. What do you mean by that? What do you mean by the word short day plant is the long night plant? which means they require the night length more when compared to the daylight. That's why we can call the, means they require more night when compared to the day. So every student, please kindly look into the pictures very carefully. Here yellow color you can see na, that is the day length. The black color you can see, right, that is the night length. If you want to understand this experiment, friends, it is very important you have to know this dotted line. What is that dotted line actually, friends? That is the critical day length. What is that, friends? That is the critical day length. Every student just pay attention towards this. Now you only keep on answering my questions here. One second. Yeah, keep on answering the questions. In the first condition, which is more? Everyone look into the diagram carefully. In the first condition, which is more? Obviously day is more. Yes or no? Of course, day is more. Now what did I tell you? Short day plants required what? Short day plant require long night. But here night length is how much? Night length is very low. Do you think short day plants will flower, friends? No, not at all. Short day plants are not going to flower. Now friends, look into the second condition. If you look into the second condition, which is more, friends? Undoubtedly night is more. Which is less, friends? Day is less. Then obviously which plants will flower? Undoubtedly short day plants will be flowering, because short day plants require the more night length. Obviously they can flower. Now every student has to understand the third condition. The first two conditions are very easy to understand. Now don't confuse. However, if the night is punctuated, everyone try to understand this particular line. If the night is punctuated by a light for a few minutes, then it will not flower. Every student first of all look into the picture carefully. Originally in the third diagram, which is more? Originally which is more? Night is more. Which is less? Day or light is less. Now 100% the short day plant has to flower. But friends, you can observe the picture very clearly. Why does the short day plant did not flower? What is the reason behind that? Short day plant did not flower. The reason, every student understand, we have punctured. What I have done? We have punctured, we have disturbed. Listen to me carefully. We have punctured, we have disturbed this particular night length. How we have punctured or how we have disturbed the night length? By flashing a light. See, in the night what I have done? I have flashed the daylight. When I flashed the daylight, what happened? Night length got punctured. Very simple concept. Friends, whenever night length got punctured, what happened? Night length is decreasing, and which length will increases? Obviously day length will increases. When there is a more day length, undoubtedly friends, short day plants are not going to get flowered. I will repeat just in 1 minute. Look into this. In the first condition, day is more, night is less. That's why friends, short day plants cannot flower, because short day plants require the complete more night length. In the second condition, night is more, day is less. That's the reason short day plants are flowered. And if you look into the third condition, originally night is more, day is less, but still it is not flowering. The reason behind that, the night length we have punctured. Puncture means we just flashed. You know, no friends, if we are moving in the dark area, suppose example, we are moving in the dark area, all of a sudden if we switch on the torch light, we will see everything, which means we are disturbing the night length. Same way here. So when we disturb the night length with the flashlight, day length increases, night length decreases. As a result, friends, short day plants are not flowering. That's a simple point. So keep it in your mind. Short day plants are long night plants. Now the second one, opposite. Long day plants are short night plants. How can I explain this? What do you mean by the word long day plant? Long day plant means it require more day length. I mean to say it require more than 12 to 14 hours of the daylight. They require very less night length. That's the reason we can called long day plants. Okay, now look into the first diagram, second diagram and third diagram. In the first diagram, which is generally more? Day is more, night is less. Then obviously which will flower? Long day plant will flower. There is no doubt about it. Now friends, look into the second situation. Which is more? Night is more. Which is less? Day is less. Do you think a long day plant will flower, friends? No, not at all. Because if the long day plant wants to flower, they always require the more day length. Since in the second diagram you can see the night length is high when compared to the day length. As a result, friends, long day plants are not going to flower. That's the simple point. Now friends, once you look into the third one, listen to me carefully. Don't confuse. If you ask me originally which is more, I think you are unable to see the diagram. Let me just listen to me whatever I am explaining. Originally which is more? Originally night is more. Which is less? Day is less. So 100% long day plant should not flower. But what happened? Every student listen. A few minutes of light during the night. What do you call, friends? We will be calling it as the night break. Because of the night break which is getting shortened, the night length getting shortened. When the night length gets shortened, which is getting increased, friends, obviously day length is getting increased. When the day length increased, obviously which will get flowered, friends? 100% long day plants will flower. So I'll summarize in 1 second. Just look into this. In the first condition, day length is more, night length is less, so long day plant got flowered. In the second condition, day length is less and night length is more, that's why long day plants did not flower. In the third condition, originally night length is more, day length is less, but what we have done is we have done the night break. What do you mean by the word night break? We have interrupted the dark period. What, friends? We have interrupted the dark period. Since we have interrupted the dark period, what happens, friends? The night length is going to get decreased, daylight increasing. As a result, long day plants can flower. So every student listen once again. Short day plants are long night plants and long day plants are short night plants. All people understand that. Now you people has to remember two important statements. If you remember these two statements, almost all the experimental based questions will be very easy to all of you. So first statement you people has to remember, red light is most effective in the night break. Next line, far red light cancels the effect of red light. Once again look into the statements. Red light most effective in the night break. Far red light cancel the effect of red light. So every student has to look into the diagram carefully. Let me explain you elaboratively. Look into the condition. In the first case, which is more, friends? Everyone has to answer my questions. Okay, friends, just give me one moment of time. By that I will decrease the image size, by that you can see the pictures carefully. Yeah, so now you can see the image. Okay, listen to me very carefully. If you look into the diagram, friends, short day plant is the long night plant. Everyone look into the picture carefully. Originally this is the first diagram, second diagram and the third diagram. Now you only tell me, friends, in the first diagram which is more? Which length is more, friends? Obviously day length is more. And which length is less, friends? Night length is less. Now we all people know very well, when there is a more night length, once again listen to me, whenever there is a more night length, short day plant will flower. But here which is more, friends? day length is more. Since the day length is more, long day plant will flower. Fantastic. Now here, if you look into the second diagram, good. Which is more in the second diagram? Night is more. In the second diagram, night is more. Which is less? Day is less. Then obviously what kind of plants will flower, friends? Undoubtedly, friends, short day plants will flower. These two steps everyone can understand very easily. Now what I want every student has to understand is the third step, which is the most important step. Don't get confused, just listen to me carefully. According to the picture, which is more actually? According to the picture, which length is more? Undoubtedly night length is more and day length is low. Day length is low, night length is more. Now you all has to get one wonderful doubt, that since the night length is more, obviously short day plants has to be flowered, but that is not happening. Long day plants are flowering. What is the reason? Because friends, every student focus, I have punctured the night light, I have disturbed the night light, I have break the night light with a flash of red light. Because friends, we have written a statement here saying that red light is most effective. Red light is most effective in interrupting the night length. Never ever forget this particular statement under any circumstances. So since red light is breaking the night length is getting decreased. Then which length is getting increased? Day length increasing. Obviously friends, the long day plants are getting flowered. So the statement, what you have to remember? Red light is most effective in night break. But what is the final statement I want you people to remember here is that, listen to me carefully, the statement whatever I am giving, whatever the function performed by the red light, that function actually cancelled by the far red light. So once again listen to me, whatever the function performed by the red light, that function basically cancelled by the far red light. Okay, every one of you pay attention towards the diagram, listen to me carefully, I'll explain twice, the entire diagram I'll explain twice, pay attention. So friends, if you look into the first one, which is more in the first condition, you can see friends, day is more. Which is less? Night is less. Since the day is more, undoubtedly long day plant will flower, there is no doubt about it. Now you look into the second condition. In the second condition, which is more? Night is more, day is less. So friends, which will flower? Undoubtedly friends, short day plant will flower, there is no doubt about it. Friends, kindly look into the third condition. What you have to understand in the third condition, originally which is more, you have to remember this, night is more, day is less actually, but still night is more, originally short day plant has to flower, but it is not happening. Why? Because I have punctured the night length with the red light, because we discussed very clearly, red light is most effective. Red light is most effective in the night break. That is the reason what is happening friends, night length getting decreased, day length starts getting increased. Since the day length is increasing friends, undoubtedly long day plants are only going to flower. Now don't get confused, look into the fourth condition, which is generally more, night is more. Which is less? Day is less. That is true, night is more, day is less. But what I have done, first I have punctured the night length with red light. If I punctured the night length with red light, which has to be increased? Daylight has to be increased, but it is not happening. What is the reason? Because friends, after the red light I have used the far red light. So friends, what does this particular far red light is doing friends? Far red light cancel the effect of red light. Very simple. Now you can ask me, sir, what exactly red light is doing here? Red light is increasing the day length. Then far red light cancel the effect of red light. Now friends, day length never increases. Which length increases? Night length increases. Then the short day plant will flower. The same experiment, once again we have exposure to the red light. See, once we exposure to the red light, long day plant will flower. Once we expose it to the far red light cancel the red light. That's why night day plant, what we can say that short day plant will flower. Once again I have treated with the red light. So once again long day plant will flower. Once again red light for red light, red light for red light. Once again here short day plant only will flower. Let me tell you one common statement to all of you. No matter, listen the statement whatever I am speaking now. No matter how many times the red light is flashed, it doesn't matter, as long as it is followed by the far red light, all the effect of red light is getting completely cancelled. What friends? Far red light is cancelling the effect of the red light, red light is cancelling the effect of far red light. That's a simple point. Once again all the students listen. Whomever did not understand, look into this once again. First condition, day is more, night is less. As a result, long day plant is flowered. In the second condition, night is more, day is less. That's why short day plant is flowered. In the third condition, night is more, day is less. But what I have done, I have punctured the night length with the red light. That's why night length decreased, day length increased. As a result, long day plant starts flowering. If you look into the fourth condition, what I have done, I have treated the red light with the far red light. So far red light cancelled the effect of red light. As a result, what happens? Obviously, night length becoming more. When the night length becoming more, short day plant will flower. So here, once again, far red light cancelled by the red light. Once the red light is more, obviously which length increases? Day length increases. Then long day plant will flower. Once again this red light I have treated with far red light. Whenever there is a far red light, it cancels the effect of red light. Then it decreases the day length and it increases the night length. Then there is a development of the short day plant. So what is the unique statement every one of you has to remember? Far red light always cancel the effect of the red light. How irrespective how many number of times you may flash the red light, if you passing far red light, it will cancel its effect. So friends, that is sensory photobiology. Okay, everyone please respond. Is it clear everyone? Is it clear? All the students please respond, please respond. Okay, perfect, perfect. Okay, I hope it's clear for everyone. Now let's start solving the questions. If anyone having any kind of a doubts, you people can ask me, friends. Okay. Okay. So friends, now we are going to solve the questions. I am requesting every student, friends, be patient. Okay. Because we are going to discuss for a very long period of time. So I don't know how much time it will take. At least we will see how many number of questions we can practice with more interest. The same phenomena happens in Tulsi plant worship in Tulsi plant with light inhibits and less flowering. Correct, Sahani. Absolutely true. That's really a good example in the nature. That's true, dear. Absolutely true. Okay. So friends, before starting the questions, I am saying please be patient. I am not at all worried whether you will give me the right answer, whether you will give me the wrong answer. I don't worry about that. I will only worry that whether you are thinking correctly or not, whether you are analyzing the question correctly or not. If you understand the question correctly, then you can analyze it correctly and you can give the answer correctly. Even though you are very intelligent, friends, if it is a part B question, if it's a part B question, spend 30 seconds, even though you are intelligent. If it is a part C question, at least keep 1 minute. Kindly do not forget it. Okay, let's start. I'll zoom. I think you can't see. Okay, I hope everyone can see it nicely. Okay. Perfect. Okay. Question number 1. Your time starts now. I want you to be little faster. Yes, friends. Yes, yes. Right or wrong doesn't matter. Answer the question. So fast. B. Sadguri has given B. Others. Okay. So friends, we all people know very well. Ethylene is the hormone. What the ethylene is the hormone will show the process called as a triple response. And I want every student has to remember, friends, it never show the triple response in every plant. It will show the triple response only in a particular seedlings. What do we call it as? Etiolated seedlings only. So what are the basic 3 processes? One we can call it as radial swelling. What do we can call it? One we can call it radial swelling. And friends, another one we basically call it as inhibition of the hypocortical elongation. What friends? Inhibition of hypocortical elongation. Then the third function it will perform is horizontal growth of epicortical. What friends? Horizontal growth of epicortical. Sometimes kindly don't get confused. Keep this point in your mind. Sometimes they will give a word called as digravitropism. Friends, digravitropism is also a correct statement only. Horizontal growth of epicortical only we can call digravitropism. So don't confuse. So decrease in apocortical elongation, it will do it. Thickening of the shoot, horizontal growth of epicortical, but rapid unfolding and expansion of leaves cannot be mediated by the ethylene. It is not the part of the triple response. Perfect. Question, I'm so sorry. Question number 2. Very good, amazing. Friends, everyone has to give the answer. Question number 2. So fast. Yes, friends, question number 2. Everyone. So we don't need to think too much here. In the phloem, in the sieve tube elements of a phloem, in the sieve tube elements of the phloem, glucose cannot be transported, friends. Actually, this particular glucose can be combined with the fructose and it is going to give rise to the sucrose. Sucrose only can be transported into the sieve tube elements. That's good. Question number 3. The quantum yield of oxygen evolution during photosynthesis drastically drops in the far red light. This effect is known as, come on friends, be patient and be active. Oh, that's great. So friends, let me define first of all what is quantum yield. Friends, how can I define the word quantum yield? Every student remember, quantum yield means, friends, number of oxygen molecules produced. Number of oxygen molecule produced per photon absorbed. Every student remember this definition. How many number of oxygen molecules are produced per single photon absorbed? That is called quantum yield. I want every student has to remember a point that quantum yield is generally constant up to 680 nanometers. Every student keep it in your mind. Quantum yield is constant up to 680 nanometers. Beyond that only, friends, it is getting drastically fall down. So that is only we can call the red drop effect. You kindly don't worry, I will provide all the crystal clear explanation and I will upload in the website. You can download it also. Okay. Question number 4. That's good. Very, very good, friends. We have already discussed this. I told you many times. See, this kind of part B questions also we will get. Faster, friends, faster. Forgot everyone. Friends, we read, right? Nitrogen plus 16 ATP molecules plus 8 protons plus 8 electrons, which is going to give rise to what, friends? It is going to give rise to 2 molecules of NH3. We read, right? Plus 16 molecules of ADP and plus 16 Pi can be released. We read this equation already. So, and I told you very clearly, nitrogen fixation is a highly energy consuming process. Obviously, they will use abundant amount of ATP. Amazing. Good. Okay. Question number 5. It's a part C question. Don't be hurry. I'll see who will be more patient today. Slowly think, friends, and answer. You know, if you analyze one option correctly, in this question I am talking about, if you analyze one option correctly, you can easily answer this question just like that. If you analyze one option correctly. Oh, someone already answered. Question number 5, what answer? Think it, be patient. C, okay. Anyone else is going to answer, friends? Amazing, wonderful. The answer will be C. Friends, you only try to understand, friends, integration of symplasmid of rhizobium in the root nuclear genome. Absolutely wrong statement. Why do we require the root nucleus? Tell me. We discussed very clearly it is occurred in the epidermal cell, root epidermis. There will be no requirement of root nuclear genome. And we all people studied very clearly that our plant root starts releasing certain flavonoids. Correct? Now we can call chemoattractants, flavonoids and betaines to attract the rhizobia. Absolutely right. Sorry, to attract the bacteria. Right statement. Activation of NOD gene in the rhizobium. Of course, once the NOD gene get activated, it is going to activate the other nodulin genes. So B is correct, C is correct, D is correct. Hence the correct answer will be C. Amazing. Wonderful. I want you to think this question and slowly and give me the answer. We already read this. Don't make a mistake, please. Today only I have explained this question. Think friends, think and answer. We discussed the question. Slowly friends, slowly, don't be confused, don't be confused. Saima Sunita has given D. What about others? Savita D. Anyone else? Okay, look into the question. Red light photoreceptors are represented by a gene family. Here what are red light photoreceptors means friends? Red light photoreceptors nothing but what? Phytochromes only, no friends. We have already discussed very clearly in the today's class only. Phytochromes can be coded by 5 genes. I told you phytochrome A, B, C, D and E. So absolutely true statement. Option A. Now look into the option B. Phytochrome C is the most prominent one. No, right? We are having phytochrome A and phytochrome B. These two are more prominent, not the phytochrome C. Hence, option B is a wrong statement. Then option C, cryptochrome 1 and cryptochrome 2 have evolved from bacterial DNA photolysis. We discussed very clearly in the morning class. Cryptochrome contain chromophore and apoprotein. I have clearly explained in morning. Apoprotein is what? A bacterial DNA. It is having a DNA photolysis enzyme, but it is do not having a DNA repair mechanism activity. Absolutely, the C statement is right. Far red light is perceived by phytochrome D. No, not at all. Hence, the correct statement is A and C. Hence, friends, the answer will be D. That's good. That's good. Friends, if anyone is getting the doubts, you can ask me immediately. I will resolve the doubts. Okay. One second. Let me show you correctly. Perfect. Question number 7. Think and answer. This question also, whatever they have asked 4 marker, you can easily analyze the answer. You can easily analyze the answer. Think it, friends. Easily you can analyze the answer. Sunita said B. Okay, what about others? Friends, you know, you can easily analyze the question. Every student answer my question. In the phloem, does the reducing sugar can be transported or non-reducing sugar can be transported? Undoubtedly. Tell me friends, everyone, does the reducing sugar or the non-reducing sugar can be transported? What kind of a sugar the sucrose is? Only sucrose can be transported. What is sucrose? Sucrose is the reducing or non-reducing? everyone. Now look into the option B. Sucrose uploading can be symplastic also, apoplastic also. Good. So sucrose is the only non-reducing sugar that can be transported. I hope every student remember. In the last 2 days back we read it very clearly and I have highlighted the 2 points. Both uploading and downloading are both loading and unloading of the sugar. What we discussed, it contains both symplastic pathway also, apoplastic pathway also. That is what we studied. Option C, the route of phloem uploading is, we discussed na, mesophyll is the major cell where the sugar synthesized. From the mesophyll cell, parenchyma, companion cell, finally sieve tube element. So C statement is right. Option D, transport in sieve tubes as per the pressure flow model. We already discussed. We can also call it as bulk flow hypothesis or Munch hypothesis. So obviously option D is also right. Hence B, C, D is right. Trust my words. If 5 questions are asked in the from plant physiology in the part C, you have to believe my words. 3 questions you can answer very easily. That is our target. Our target is not answering 5 questions. If 3 part B questions they have asked, at least we should be in a position to answer 2 or at least 1. That is our target. Okay, good. Next. Question number 8. Think and answer, friends. Think and answer. Don't be hurry. Everyone, slowly. Check Sumaya, check dear ones, check it slowly. Don't be hurry. Friends, okay, what about others? Other. We have discussed many times. From the P680, who is the first electron acceptor? From the P680, pheophytin. From the pheophytin, plastoquinone A, plastoquinone B, plastocyanin, cytochrome B6F and P700. So obviously option A, look into this. They said P680, pheophytin. I'm so sorry friends, one second, let me write here, let me write, I'm so sorry. First P680 followed by the pheophytin, then followed by what friends? Quinone A, quinone B, immediately cytochrome B6F, last one is the plastocyanin. But if you look into the option A, P680 correct, pheophytin correct, quinone A, quinone B correct, but this is wrong, right? After the cytochrome B6F, plastocyanin will come. Hence option A is wrong. Option B, P700 followed by the phylloquinone. Everyone remember, what did I tell you? All of you, phylloquinone called as A0. Anyway, they did not give the A1. So P700, phylloquinone war are A0, iron sulfur SA, SB, SX, FD. Absolutely true. Option B is right. Option A is wrong. Option C, friends. One second. What is wrong? I'm sorry, friends. I think I made it wrong. I'm extremely sorry. I made it wrong. Sorry. I made it wrong. Okay. Just give me 1 minute. First, let me write. Because of this, I got confused. P680, pheophytin, plastoquinone A, I'm so sorry, I made it wrong. Plastoquinone B, then cytochrome B6F, plastocyanin. This is absolutely true. There is no doubt about it. Then when you look into the P700, what you have to understand, P700 I told you now, after that A0, A1, A0 we can call modified chlorophyll A, A1 we can call phylloquinone, then followed by iron sulfur X, iron sulfur A, iron sulfur B, then followed by FD, then followed by FNR, then finally NADP plus. This is the real electron scheme. So if you look into the option B, P700 phylloquinone, FAS, FAB, FESX, FD. No, listen to me everyone carefully. After first is iron sulfur X, after that iron sulfur A and iron sulfur B. So undoubtedly friends, option B is also wrong option, option A is also a wrong option. Now if you look into the option C, P680, pheophytin, quinone A, quinone B, cytochrome B6F, plastocyanin, P700. Option C is absolutely right. Option D, P700, anyway they did not given the A0, they have only given A1. A1 only we can call phylloquinone, absolutely right. Iron sulfur X, right. Iron sulfur A, iron sulfur B, FD, FNR. Anyway they did not give the FNR. Absolutely D is right. Hence the correct answer will be C. Correct answer will be C. Right, whatever you all people given is absolutely right answer. Perfect, perfect. Okay, just a moment. Okay. Yes, friends, question number 9. Okay, first you look into this option, look into the question, then I will show you the options. Otherwise, just give me a moment. I will write the options here itself. By that you people don't get confused. So option A is 1. No, friends, just take a lot of time to write it. Why to waste the time? Okay. First you look into the question, then I will show you the options. Writing is waste of a time. A, tracheids, B, vessel elements, C, sieve tube, D, sieve cells. I have explained this to all of you in a specially in 2 days back also. I told to all of you to read and remember this. Tracheids, vessel elements, sieve tube, sieve cells. Okay, now kindly look into the options. If I make it little less zoom, you can't see, that's the problem. Okay, if you can see, that's good now. Let me know if anyone is unable to see it, I will show you. It's a very easy question. Friends, I want you to look into the question carefully. I have explained you very clearly, friends. 4 or 5 times I have shown a table to all of you, and I told you, friends, this is important. You have to remember this particular table, and I told to all of you, don't be hurry. Okay, everyone listen. Tracheids, I have clearly told you, tracheids are not highly modified. Since they are not highly modified, they are present in the gymnosperm. And if you look into the vessel elements, they are very highly modified. So obviously they are present where? In the angiosperms. Very simple point. And if you look into the sieve tube is not highly modified. So sieve tube is highly modified. So it will be present in the angiosperm. And if you look into the sieve cell is also modified, but when compared to the sieve tube, it is little less modified. So it is present in the gymnosperm. Then how does the answer will be C, friends? Answer will be A. Answer is not C. Tracheids present in the gymnosperms. Vessel elements present in the angiosperm. Sieve tube present in the gymnosperm. Then how does the answer will be C, friends? Answer will be A. Answer is not C. Tracheids present in the gymnosperms. Vessel elements present in the angiosperm. Sieve tube. Every student remember, sieve tube. Tube is modified. Friends, unnecessarily because of a small thing you made a mistake. Very small it is. Sieve tube. Tube is modified. So highly modified always present in the angiosperm. Sieve cells, they are modified, but when compared to the tube they are little modified. So that's why they are present in the gymnosperm. Friends, please kindly don't confuse. Don't confuse unnecessarily. Tube is highly modified angiosperm. Cell is little modified gymnosperm. Don't confuse, please. Next one. Question number 10. Part C. Easy question, friends, so you can answer this. Just think it correctly. Think it correctly. Slowly. Oh, Jayashri already given the answer. Check dear, check. Don't be hurry. Amazing. Okay. Friends, look into the question. The question says that following are certain statements regarding terpene class of secondary metabolites. Isopentenyl diphosphate and its isomer combined to form a terpene. Of course, true, right? IPP. IPP is what carbon? 5 carbon. I hope every student remember. And what is the isomer of the IPP? I hope all of you remember. I told you the isomer of IPP is what, friends? DMAPP. DMAPP is a 5 carbon. IPP is also 5 carbon. Together they will give the monoterpene, which is an active form of terpene. Obviously option A is right. Now look into the option B. Option B says that diterpenes are 20 carbon. Undoubtedly. What is the best example we people studied? GGPP. Ziranyl diphosphate or ziranyl biphosphate. Right statement. Option C says that all terpenes are derived from the union of 4 carbon. Absolutely right. Wrong, friends. It is derived from how many carbon? 5 carbon element. So friends, option C is a wrong statement, and we discussed very clearly pyrethroids, which are acting against the insect, which is present in which plant? Which is present in the chrysanthemum. I have given the example of chrysanthemum and I have explained. So pyrethroids are monoterpene. So friends, D is right. Hence option A is correct, B is correct, D is also correct. Hence the correct answer will be option B. That's good, amazing. This question I want you to think and answer. Think and answer this question. Question number 11. Don't be hurry, please. Come on friends, be patient. Try it. It's not a difficult question, but the statement you have to remember here. Try it, no problem. Okay, anyone else? Only Saima given the answer. Others, right or wrong, say it friends, right or wrong, say it. Okay, so friends, we all people studied what is DELA. DELA protein is the suppressor. It can act like a negative regulator. It is acting as a suppressor of the gibberellic acid. If they, imagine friends, listen to me carefully. If there is a no gibberellic acid in the plant body, listen to me. If there is a no gibberellic acid in the plant body, DELA repressor get active. Once the DELA repressor get active, then the DELA repressor suppress the gene expression. When the DELA repressor is getting activated only in the absence of gibberellic acid. Now see here, option A says that binding of gibberellic acid bound repressor to the promoter of the DELA domain containing GRAS protein gene and blocking its expression. First of all friends, binding of gibberellic acid bound repressor to the promoter of the DELA domain. It is not at all possible. Option A is completely wrong option. Why gibberellic acid will go and bound to the repressor friends? Gibberellic acid will go and bound to its receptor. This is the question asked in the 22 also, right? 2020 November paper. What was that? GID1. GID1 is the name of the receptor to which gibberellic acid will come and attaches. Gibberellic acid never bound to the repressor. Statement A is completely irrelevant and wrong statement. It never bound to the receptor. Option B, binding of the gibberellic acid receptor complex to the GRAS. You know, GRAS is an important protein that is also involved in the regulation of the gibberellic activity. The moment, every student listen to me carefully, the moment gibberellic acid come and associated with the GID1, immediately what happen? The GRAS protein can go and added by the ubiquitin molecule. Once it is added by the ubiquitin molecule friends, obviously it will undergo the process of proteasomal degradation by 26S proteasomal complex. That is what exactly friends the gibberellic acid will do. So B is right, C is right. And what is the role of microRNA in the gibberellic acid expression? In the gibberellic acid expression, there is no role of microRNAs. No. Why we are going to regulate the RNA level here? We are not regulating anything with respect to the RNA level. Absolutely wrong statement. Correct statements are B and C. Hence the correct answer will be B. It's a very simple and very direct question. You have to think and answer. Upon multiple times revision, you can easily remember. Don't get confused. Gibberellic acid associated with the GID1 and DELA protein can act as a regulator of the gibberellic acid or suppressor of the gibberellic acid. Remember that point. Okay. Question number 12. This is also kind of tough question. Not greatly tough. Just think and answer. This is friends EIN2. This is called EIN2, transcription factor. Priya someone asked, Priya, GRAS is the protein which is activated by the gibberellic acid. Now once the GRAS protein get activated, GRAS protein will stimulate the ubiquitin system. Then ubiquitin will come and attaches to the DELA protein. Then the DELA protein degraded by the 26S proteasomal complex. So dear, the exact function of a GRAS which can bring the response of gibberellic acid by removing or by inhibiting the delta repressor which is associated and blocking the gibberellic acid expression. GRAS protein is actually required for the delta protein degradation with the help of ubiquitin system, and GRAS can be activated by gibberellic acid. You got it? Yes, yes, yes. Question number 12. You can answer this question also very easily, friends. We have discussed this. Oh, everyone answer. Okay, look into the given question. The question says that ethylene is an important plant hormone that regulate several aspects of plant growth and development. Some statements are given below in relation to the ethylene signaling pathway. Every one of you please kindly look into the option A. Option A says that unbound ethylene receptor work as a positive regulator. How does that possible? Without the presence of a ethylene, how plant can show the positive response? Option A is absolutely wrong option. I have told you very clearly, there are more than 5 ethylene receptors were identified in the Arabidopsis thaliana. I hope you remembered. And I told you, in case of a Vorija sativa, there is one ethylene receptor was identified. So option B says that there are more than 2 ethylene receptors are known to date. Absolutely true statement. There are more than 5 was identified. They said more than 2. Right statement, option B. Option C, carboxy terminal off of the ethylene receptor ETR1 contains a domain homologous to the histidine kinase. How many number of times I have reminded you, even in the unit 4 also. We are having different type of receptor kinases, right? Receptor tyrosine kinases, receptor threonine kinases, receptor serine kinases, receptor histidine kinases. So serine kinases and histidine kinases, they basically present in what, friends? Plant. ETR1 and CTR1 domains. I told you, right, ethylene can show the hybrid 2-component signaling system. I hope you remembered, which is ETR1 domain, and it is having another domain called CTR1 domain. So the carboxy terminal half of the ethylene receptor ETR1, it resembles the histidine kinase activity. Absolutely right it is. E1N2 encodes a transmembrane protein. It is wrong statement. Friends, what is the function of a E1N2? E1N2 actually acting as the transcription factor, which will go and stimulate various type of genes which will involved in showing the response of ethylene. But they said E1N2 can encode for transmembrane protein. Not at all. It never involved in the it is not involved in coding for a transmembrane protein. It is a transcription factor, enters inside the nucleus. Option D is wrong. Option B is correct statement. Option C is also correct statement. Hence the correct answer will be B. Perfect, perfect. Let me know if anyone having any kind of a doubts, please ask me. Question number 14. The 13th question, it was not completely typed, it's a wrong question. Question number 14. People are leaving. Come on friends, be patient. It's very simple question, I want you to answer. Okay, anyone else? Okay, so friends, we all people commonly know that there are two type of genes are there. One we can call the common nod genes and host specified nod genes. Correct na? And I told to all of you, if the common nod genes and host specified nod genes wants to be activated, we require one very important gene. What is that gene? We call it as a nod D gene. So first in the bacteria body, what gene get activated? Nod D gene get activated by the chemoattractants like flavonoids and betaines. Once the nod D gene get activated, this is the gene which can code for common nod gene and host nod genes. Host specified nod gene. This is the promoter region. What do you call it, this promoter region? We can call nod box. To this nod box, who will come and attach us? Nod D will come and attach us. The moment nod D will come and attach us to this particular nod box, there will be an activation of the common nod gene also, host specified nod gene also, then the nitrogen fixation occur. Fantastic, amazing. The correct answer will be B. Very good. It's a direct and straightforward question. Question number 15. Question number 15. See, what about others? Come on, everyone. See friends, we discussed that cytokinin can show the two component signaling system. Ethylene can also show the two component signaling system. But in a given question, they did not give any kind of the what we can say that ethylene in the option. Then undoubtedly what you will choose? You will choose the cytokinin is the correct answer. Amazing. Question number 16. Okay friends, I want you to remember this. This G loop, anyway, this we will read in the evolution also actually. Question number 16, we will read in the evolution also. (15) is C. That's right. Yeah, right. Absolutely right. B. Yeah. Simon, what is your answer for B? Cryptochromes. Okay, so friends, listen. The answer for this is Jeet loop family. Okay, Jeet loop family is a type of a specialized. See, friends, they did not ask you. I don't know why you have chosen the answer separately. Cryptochrome and phototropins. These two are the different type of photoreceptors. Anyway, I will agree with you. This particular phototropins having a key role in the circadian rhythm. I will agree with all of you. But that is not the question asked. Which of the following photoreceptor play a role in the day length perception and circadian rhythms together? Jet loop family is a type of protein which can sense blue light flavoprotein. Okay, no problem. Whomever made it wrong, remember the jet loop is a blue light sensitive or blue light sense flavoprotein which is involved in maintenance of the circadian rhythms in the plant body by maintaining the day length perception. Answer will be A. Jet loop. It's not cryptochrome, it's not a phototrope. Please kindly don't confuse and remember this. In the evolution, when we are reading the circadian rhythms, we will remember this. We will read this. Keep it in your mind. Jet loop family proteins are blue light sensitive blue light sense flavoprotein involved in the day length perception and circadian rhythm? Answer is A. It's not B and C. Don't confuse with this. Okay. Question number 17. Don't confuse, don't confuse. Slowly, friends, slowly think it, think it, think it. You're not thinking. Okay, that's good. The question number 17, the answer will be D. Same, right? After P680, immediately plastoquinone A, plastoquinone B. Anyway, they just commonly given plastoquinone, followed by cytochrome B6F complex, followed by plastocyan. Fantastic. Answer will be D. This question number 18, it is not related here. It is from unit 5. Okay, we will read in the unit 5. Question number 19. Think and answer. It's a very big question and very important question. Come on, be patient, friends. You have to choose the right statements. See, imagine, friends, how many times they are asking the signaling questions in the part C. Question number 19. Okay, friends, look into the question. The following statements are made to describe auxin signal transduction pathway from receptor binding to the physiological response. I have explained it to all of you very clearly. If the auxin want to show the response, what we required? We require a transcription factor called as ARF, auxin response factor. We discussed many times, in the absence of auxin, this ARF associated by a inhibitory gene. What do you call that inhibitory gene? Auxin IAA gene. In the absence of auxin, this auxin IAA inhibitory gene, they block the ARF. But whenever the hormone auxin, indole acetic acid, will come and associated with its receptor, what is the name of the receptor? Also I told to all of you. The name of the receptor is what? TIR1. That is also the question asked. Auxin come and associated with the TIR1 receptor, immediately there is an activation of ubiquitin pathway. Now ubiquitin molecule will come and add it to the auxin IAA, and it will undergo the proteasomal degradation by the 26S proteasome. Then ARF become free. It will go and associated with the particular sequence in the DNA. Then it show the response of auxin. That is what we people study. We read in the signaling clearly. Now look into the option A. Auxin response factors ARF are nuclear proteins that bind to auxin response element. Of course true. Where the transcription factors will associate? Transcription factors associated within the nucleus. What is their function? They are the one which will regulate the transcription of the hormone. Absolutely A statement is right. Option B, auxin IAA proteins are secondary regulators of auxin induced gene expression. Binding of auxin IAA protein to the ARF get blocked. Absolutely right statement. Option C, auxin binding to TIR1 receptor or AFB, anything is same. Promote the ubiquitin mediated degradation of auxin IAA. Right statement. Auxin never ever come and bind to the ARF. That is wrong statement. Auxin never associated with its response factor directly. Auxin always stimulate its response factor. Always keep that particular point in your mind. So option D is wrong. Option A is right. Option B is right. Option C is right. Hence the correct answer will be A. Absolutely fine. Okay. Question number 20. Very good friends. Very good. Option A is correct. Fantastic. Question number 20. I'll write the options here itself. Option A, B and C. Option B, A, C and D. Option C, B, C and D. Option D, A, B and D. Friends, they are asking correct statements. Come on, be patient. More questions to be solved. So fast. Think it correctly, think it correctly, think it correctly. Very good, very good, very good. So the correct answer will be what, friends? C. Because friends, we can easily eliminate this option A. The reason PS1 reaction center and PS2 reaction centers are not uniformly distributed in the granule and stromal. We have discussed very clearly. If you look into the PS1 present in the stroma exclusively. PS2 is present in the grana exclusively. Option A is wrong. So of course, if you come to know that option A is wrong, you can easily come to know it. I mean, so you can easily guess it. Anyway, we know that the electron donor for the P700 is what? Plastocyanin. And from the P700, who is going to accept the electron? It's not A, it's a A0. I told you now. Then the core antenna and P700 bound to PSA and B. Absolutely right. B is right, C is right. Cyclic electron transport flow occurring from the reducing side of PS1. We already discussed, right? Cyclic photophosphorylation, we can only produce the energy equivalent ATP. We never produce the reducing equivalent, that is the NADPH. So D is also right. Hence the correct answer will be C. That's good, amazing. Question number 21. We discussed this, friends. We discussed this. Combination of statements we have to read here. Twenty-one. Okay, friends, please ask me if you have any doubts. We all people know very well, friends. Suppose example, this is the Rubisco. Rubisco is an enzyme. You only tell me, friends, does the Rubisco having two active sites? No, right? Every single enzyme, every enzyme having only one site, one active site. Only to that one active site substrate will come and attach us. Very simple point. So option A says that the active site on Rubisco for carboxylation and oxygenation are different. No, there is only one active site will be present. One of the photorespiration, one of the steps in the photorespiration is conversion of glycine into serine. Where does the glycine is going to be converted into serine, friends? Of course, in the mitochondria. So option B is the right statement. Option C says that 50% of the carbon lost in the chloroplast due to oxygenation is recovered through the photorespiration. No, the pathway of photorespiration involves CPM, chloroplast, peroxisome and mitochondria. So option B is correct, option D is correct. Hence the correct answer will be C. B and D. Very good, amazing, friends. Good, very good. Think and answer. Question number 22. I'll write the options here only. Option C and D. Option D, A and D. A and D. Slowly, friends, think and answer. Slowly think it. Come on, be active. See. Give me the correct answer, friends. Think it, please think it. Okay, 22. What is the answer? Most of the people say that C, D. What is this? Okay, anyone else? 22, friends. It's a easy question, friends. Look into the question carefully, all of you. The question says that, Deepika, how D, dear? You only tell me how it will become D and A. Every student look into this. All the students answer my question. pentose phosphate formed by the photosynthesis or hexose phosphate formed by the photosynthesis? Are we going to produce the glucose? Are we going to produce the ribose? We are going to produce the glucose, right? Glucose is how many carbons? Everyone, glucose is how many carbon? 6 carbon. But then why you are answering pentose phosphate? Then option A is wrong, right? Then option A is wrong. You can eliminate option A also. You can eliminate option D also. It's not a pentose phosphate. In the photosynthesis, we will produce the glucose, friends. Glucose is how many carbon? 6 carbon. Don't forget that. Option A is wrong. It's a very simple common sense question. No student is observing this word. No student is observing this word. Pentose phosphate. Why do we produce the pentose phosphate in the photosynthesis? We produce glucose. This is wrong. Option B, carbon stored as a starch exists the chloroplast at night primarily in the form of maltose, right? And it is converted into sucrose in the cytosol. I have told you many times, sucrose production where it will occur in the cytosol, and starch where it is synthesizing in the chloroplast stroma. We all know that option B is right. Sucrose is sucrose is synthesized in the cytosol. Option B is right. Option C, during the short distance transport, sucrose moves from producing cell. What are producing cells? Nothing but mesophyll cell in the vicinity of the sieve elements in the smallest veins of the leaf. This statement is absolutely true statement. Friends, you know these lines, I want you to listen. These lines are exactly the lines of the what we can say Lincoln Tay's book. You know, no friends, this is the leaf, na? Leaf contain veins like this, small veins. We can call veins all this. Through that veins only, friends, sugars will be passed in the sieve element. Option C is 100% true. There is nothing wrong in the option C. Kindly don't get confused. An example, even though you feel C statement is a wrong statement, even though you feel you are confused with the C statement, with B statement you can easily answer the C statement. B is absolutely true, where the sucrose is going to be produced in the cytosol only. Option C is true, friends. The sugars will be passed in the small vein of the sieve elements. Don't confuse with that. C is right statement. These are all the exact lines of Tay's book. Option D, in the process of phloem loading, sugars are transported into phloem parenchyma. I don't know how you can choose. Option D is the correct answer. Tell me, if you look into the option D, why does the sugar transported via phloem parenchyma, friends? We all people read, na? Sugars can be transported via sieve tube element. They are asking a statement transported. How the food move through the parenchyma? Just from the parenchyma it enter into the sieve tube element and companion cell. It never ever transported. Transport occurred only through the sieve tube element. Friends, I have explained this concept more than 30 minutes continuously. How does parenchyma cells can involved in the movement? Only sieve tube. So you can make this statement also wrong. So the correct answer is B and C. Hence the correct answer is B. Please let me know if anyone having any doubt. I will re-explain, please. That's okay. I hope everyone understand it, friends, correctly. Yes. Next one. 23. Deepika, no sorry dear. Why to say sorry? That's cool. No sorries. I'm just saying you got confused between the pentose and hexose. That's all. Yes, question number 23. What about others? Come on, friends, please be active, please. Jayashri, so fast, dear. Sunita, think it, dear. What's wrong? Okay, so we read now friends, these three we read in the alkaloid. I have shown you one table, right? Indole alkaloid, tropane alkaloid, pyrrolene alkaloid. We read now friends, morphine, codeine, thebaine, heroin, all these we read, right, in the table. What about the lignin? Lignin is, we know, right friends, lignin is belongs to the phenol. It's not belonging to the alkaloid. Na? Indole is the major class of alkaloid, dear. Okay, don't confuse, don't confuse. Hence this answer will be A. Good. Question number 24. That's a good statement, but very simple statement. I don't know friends, you remember or not, I have drawn one diagram also and I have explained it to all of you. While explaining this, I have drawn root epidermis, cortex, cortical cells, and I have drawn endodermis, stele, then followed by the xylem. Okay, everyone tell me what is the answer for 24. Okay, think it. Hope all of you remember. Okay, so friends, all the students listen to me. This is what friends, every student listen to me. This is the root hair. Nothing but what friends? Epidermis. Correct na? Epidermis contains, after epidermis what is there? Cortex. Cortex contain what cells? Cortical cell. After that endodermis. After that xylem. This is what we people study. Now what is symplastic pathway? So through the plasmodesmata, inside the cell is the plasmodesmata. Through the cell wall intercellular spaces, is a apoplastic pathway. I told you very clearly. So water travel across the root cortex via plasmodesmata. If water crossing plasma membrane on the surface, obviously it is apoplastic. That is extracellular spaces. Since they asked symplastic, the correct answer will be B. Very good. Okay. Question number 25. We have discussed in the photosynthesis. Come on friends, please be active. Okay, question number 25. What will be the answer, friends? Okay, fantastic. Question number 25. The herbicide dichlorophenyl dimethylurea is an inhibitor of. We discussed now, friends. It is actually block the electron in between P680 to the P7. It's a direct question, friends. The answer will be B. This question we have already done. It's got repeated. It's repeated. Done it is. Okay. Question number 27. See everything. Think it, friends, think it and answer. Okay, what is the answer for question number 27? Think it, friends, think it, friends. (27C). Okay. Okay, what about others? Someone given B, someone given C. Okay, what's the answer? Okay. Anyone else can give the answer? Okay. Look into the question carefully. The C4 carbon cycle CO2 concentrating mechanism evolved to reduce the photorespiration. The following are stated as important features of the C4 pathway. Everyone look into this. Option A. The leaves of C4 plant have Kranz anatomy. What do you mean by the word Kranz anatomy? We people studied, friends. Kranz anatomy means it contain mesophyll cells also, friends. Kranz anatomy contain bundle sheath cells also. We people know it. Then option, so friends, undoubtedly option A is right option. Option B, first who will fix the carbon? We all people know, right, friends? In case of C4 plants, rubisco, where it is present in the bundle sheath cell. So what will be the major enzyme in case of a C4 plant in terms of mesophyll cells? Pep carboxylase. What is the function of pep carboxylase? It combines the PEP with the carbon dioxide and it is giving rise to oxaloacetic acid. So in the peripheral mesophyll cell, atmospheric carbon dioxide is fixed by the phosphoenol pyruvate carboxylase yielding 4 carbon acid. Right statement. Now friends, every one of you please look into the option C. Option C says that in the inner layer of mesophyll, NAD malic enzyme decarboxylase is present. No, right? Where does the NAD malic dehydrogenase enzyme is present? It is present in the bundle sheath cells. It is not present in the mesophyll. Hence the C statement is wrong. Now you look into this. D. Carbon dioxide again is refixed through Calvin cycle in the bundle sheath cell. Why? Because in the bundle sheath cell, friends, rubisco is present. Hence what will be the answer? Option A is right, option B is right, option D is also right. Hence the correct answer will be C. There is no doubt about it. That's really good. Great, 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 great. Question number 28. Oh, so sorry, friends. I'm so sorry. So sorry. Okay, friends, 28. What will be the answer? (28) is B. Okay, all the secondary metabolites are constitutively produced in all cells of a plant during its entire life. You know, right? I used to highlight this kind of statements only in the classroom. None of the secondary metabolite is Sadhguru, listen here. None of the secondary metabolite never ever released in the plant body every time. See, you only tell me, every one of you, does our body produce the antibodies every time? No, right? When our immune defense mechanism is going to produce the antibodies, whenever there is a entry of antigens. Otherwise, our body never produce the antibodies. Same way, friends, in case of plant also, plants not synthesizing the antibodies, secondary metabolites every time. Then option A is wrong statement. Option B, they serve as the signals to plant survive adverse condition. Absolutely true. They may be volatile. I told you this point also very clearly. Some of the secondary metabolites are volatile. You take an example of monoterpene, they are volatile. You take an example of triterpene, they are non-volatile, which means some secondary metabolites are volatile, some of the secondary metabolites are not volatile. Hence, they have given the statement that they are volatile, then the statement is right. They contribute to the flower color. What is the best example you can take? Anthocyanin, which is a type of phenol. What is phenol? Secondary metabolite. So D is also right statement. Option B is the correct. B, C, D. None of the secondary metabolite never released by the plant body every single time. Always remember that. That's a good point, friends. That's a good point. Okay. One second. I'm so sorry. Yes. Question number 29. Hope every one of you will remember this. So fast. I have highlighted this while discussing. Okay, everyone. Question number 29. Sunita, how dear? How abscisic acid it is? See, I told you, na friends, you forgot. It's there in the PPT. We read the cycle. We can call it Ang cycle. I told to all of you first. Methionine combined with adenine. Then what do you call? What we get? We will get Edomet, adenosyl methionine. And I have clearly told to all of you, adenosyl methionine can be treated by an enzyme called as ACC synthase. Then what we are going to get when the Edomet can be treated by an enzyme called as ACC synthase, it is going to give rise to the ACC. Correct, na? Now what is that ACC? Aminocyclopropyne 1-carboxylic acid. Later ACC giving rise to the ethylene. Hence the correct answer will be, friends, C, ethylene. Don't confuse, please. Question number 31. It's also very simple question. Very simple question. Friends, please kindly look into the question carefully. What they have asked you, look into this. They have asked which is incorrect, not the correct, friends. You have to give the answer which is incorrect. So fast. 31. Friends, all the students listen, remember this all the time. DCMU is basically block the photosystem 2, whereas paraquat can block the photosystem 1. Absolutely true. Then but see here, option A is wrong. They have given opposite. Option B is wrong. Option C is wrong. Option D only the correct option. Hence the incorrect statements are A, B, C. That's good. See, I want you to know this, friends. Question number 32. I did not explain the jasmonic acid, but it's very important, friends. 32. What's the answer? Okay, so friends, kindly don't get confused. Whomever doesn't know the answer for 32, just listen. Always remember these two lines. MYC2. Always remember MYC2 is the positive regulator. MYC2 is the positive regulator. JAJ is the negative regulator. JAJ is the negative regulator. MYC2 is the positive regulator. Now friends, every students understand. Suppose example, in the plant body, what is not there? Jasmonic acid is not there. What happens? When the jasmonic acid is not there, do you think jasmonic acid response can be shown? No, there is no way jasmonic acid response cannot be shown. Now friends, JAJ protein in the absence of jasmonic acid get activated, which is a negative regulator, suppress the positive regulator. When it suppress the positive regulator, what happened? Obviously friends, the jasmonic acid action will be completely repressed. So this JAJ bind to the MYC2 and it never transcribe, it never always repress the gibberellic acid dependent genes. Never forget that. Hence the correct answer will be A. Remember these 2 points friends, you can easily remember whomever did not know this. MYC2 is the positive regulator, JAJ is the negative regulator. We all people know that negative regulator is going to be active only in the absence of the hormone. If there is a presence of hormone, negative regulator is not going to be activated. So in the absence of hormone, JAJ negative regulator block the positive regulator, hence there is no response. Simple point. Hence the answer will be A. That's good. 33. See, I am requesting all of you, don't confuse. Think and answer this question, friends. Parthenocarpy. Okay, think friends, think friends. I have explained it to all of you. No, you're forgetting friends. Slowly, slowly. Friends, 33 answer. What about others? Mrunalini, what is the answer? Others, Priya. Others, Deepika, what will be the answer? 33. See, the 33 answer will be parthenogenesis. See friends, I told you hormone auxin involved in the parthenocarpy, hormone gibberellic acid also involved in the parthenocarpy. Anyway, gibberellic acid is exclusively responsible for stem elongation and it is involved in the alcohol production, exclusively in the malt production. It cannot be involved in the parthenogenesis. Friends, what is parthenogenesis? It's a type of asexual reproduction. Why the hormone is involved in this particular asexual reproduction? Not at all. Hence the answer will be C. That's good, that's good, that's good. Great. Okay. Think and answer this question. Don't be hurry. This question is easy. I want you people to remember where exactly the Casparian strip is present. And once again listen, they are asking which is not correct. They are asking, friends, you did not see the question. I am requesting all of you look into the question. They are asked which is not correct. Not looking into the question. Look into the question, friends. Look into the question. They are asking not correct. Slowly, slowly, slowly. Don't be hurry, please. Don't be hurry. Please be patient. Please be patient. See friends, I will really disappoint if you answer this kind of questions also wrong. We are reading about the Casparian strip from our 10th standard. They did not ask which is correct, friends. They asked which is not correct. I don't know why most of the people are giving the answer as A. It breaks the continuity of water movement through the apoplast pathway. That is its function, but that is not the question. Which is not correct, they are asking. It's a correct statement. Casparian strip is the one which Casparian strip contain a chemical. What is that chemical? We call it as a suberin. Suberin is the chemical what? It's a lipid. Now it blocks the movement of the water molecule. We all people know that this is right statement. Everyone listen. Where does this particular Casparian strip is present? It is present on the endodermis. It is formed in the growing part of the root. That cannot be. This is the wrong statement. It is formed several millimeters or several centimeters behind the root tip. Right statement. And what did Casparian contain? Suberin, as well as it is also contain lignin. Hence the wrong statement which is not correct is the B. Don't confuse, don't confuse. Correct dear, it breaks the simplest pathway. Right. For this the not most appropriate, this is only wrong statement. It is formed in the growing part of the root. See, endodermis is actually present on the Casparian strip. In the chapter also I told you now, because of the Casparian strip only, the apoplastic pathway skip it, and once again water molecules re-enter into the symplastic pathway. Why? It contains suberin, it never allows the movement of the water molecule. So don't think that the Casparian strip is very long strip. It's a very small strip. So C is right. Option D is also right. Sorry. Option B is wrong. Option C and option D. It breaks the continuity of water movement through the apoplast. That is the right statement only, no Sumaya? It's right? Okay, listen, for all of you I will explain once again. This is the epidermis. These are all cortex. I shown you the diagram also, no dear. And after the cortex, what is there? Endodermis. Example. Here Casparian strip is there. You know now, water moves like this, move like this, move like this, and it will come here. Do you think it will move? No, right? Now the water movement got stopped. It break the continuity of the water. Whatever the water, it has to move like this. Look into the arrow what I am showing. Water has to move like this, but water start moving down because it is entering into the symplastic pathway. We discussed very clearly. Casparian strip convert apoplastic into symplastic. So it breaking the movement of water. Absolutely right statement. They did not ask which is correct. They asked which is not correct. I hope you understand dear correctly. Let me know if you did not understand. I will re-explain in other way. Got it? Okay, good. Question number 35. Think and answer. Think and answer. This is also very good part B, part C question. I will write the options here, friends. B and C only. Option B. A and B only. Option C. C only. Option D. A and D only. Friends, it's very logical. It's not logical, you have to remember. I want you to spend 1 minute. Spend on the question at least 1 minute and try to give me the answer. Think it, no problem. Think it, friends, think it. Please be patient. I really appreciate all your patience. It's a kind of no. They're asking correct statements. Sumaya, what is your answer? D for which question? 35. Jaya given A. Sunita also given A. How many times I have told to all of you, friends? Okay. Saima, answer my statement, dear. Answer the statement. Does any kind of a alkaloid, any kind of a Mrunalini, you also answer. Any kind of alkaloid or any kind of a secondary metabolite, will it be stored in the toxic form in the plants? Savita, tell me, any kind of a secondary metabolite, will it be stored in the toxic form in the plant? How Savita, Sumaya, Saima, how Mrunalini? Sumaya has given correct answer. How it can be stored in the toxic form? How many number of times I have told you? Forget about cyanogenic glycoside. Every secondary metabolite, whenever it is present in the plant body, keep it in your mind. Whenever they are present in the plant body, they are in the inactive form, non-toxic form. When the plant got crushed, when the plant got damaged, then only non-toxic form is going to be converted into the toxic form. Then how does the option C will be the correct? Cyanogenic glycoside are produced and stored in the toxic form. Never ever. If it is stored in the toxic form, plant will die, right? Obviously plant will die if it's in the toxic form. Slowly, friends, think it. Now look, option C is wrong statement, friends. Option A, glucosinolates are synthesized by the elongation of the length of side chains of their precursor amino acid. I have explained it to all the students. The last one is what? Alkaloid, I mean to say nitrogen containing compound. The last category of the secondary metabolite. What is the line I have used in this PPT slide? Once again, look into the PPT slide. There is a line written that all the nitrogen containing compounds are derived from the common amino acid precursors. That also we discussed. So the statement A is absolutely true statement. Why the A statement is a wrong statement? A is absolutely right. All the nitrogen containing compounds are derived from the amino acid. We discussed, right, morphine, codeine, thebaine, heroin, they are all coming from what? Indole. Means an amino acid tryptophan. So all are derived from amino acid. Option A is absolutely right. Option B, all terpenes produced in the herbs and spices are sticky, oily liquids which reduce the palatability of these plants. I have explained it to all people. They never ever reduce the palatability. Secondary metabolites never ever reduce the economic importance of a plant body. Secondary metabolites never ever reduce the growth, never ever reduce the development of the plant body. They never affect the plant growth. Hence, but they said that they reduce the plant edibility of the plant. Option B is wrong. Option C is wrong. Option D, the defense molecules, alkaloid or their precursor gathered from plants and used by some insects for their own protection against predator. This is a mechanism. This is the defense mechanism used by this. So this point I can conclude, I can agree with all of you, this point is a new point, but you have to remember one common statement. Whatever the insect that will take the alkaloid or their precursor, they will use to kill the other organisms. Means they use it to protect themselves from the attack of the higher organisms also. Option D is also correct. I can agree option D is a difficult statement, but you can easily eliminate the option B. If you eliminate the option B, you can come to a conclusion, that is D. Option A and D. A and D are the correct statement. I hope it's clear for everyone. Let me know if you have any kind of a doubts. I will re-explain. Please let me know. Okay. Question number 37. Slowly friends, slowly. Think and answer. Good, very good. Come on, don't sleep. We understand that plant can show many kind of a defense mechanism. Correct na? We read that what is the first defense mechanism? Photochemical quenching, releasing in the form of a heat. Releasing excess photons in the form of a heat. Second one is what friends? Scavenger molecules like what we can say that carotenoids, ascorbate, all this. Then finally what happened? If these two defense mechanism got failed, immediately what happened friends? Excess photon will directly go and damage the D1 protein. I hope every student remember, once this D1 protein get damaged, how can we repair this? Friends, we all discussed we can repair this by de-novo-synthesis process. Very good. Hence the correct answer will be D. Amazing. Great. Question number 38. Slowly friends, think and answer. Don't get confused. They are asking friends, look into the statement. They are asking mitochondria to peroxisome. Okay, everyone given the answer. Oh, that's great. So question number 38, what will be the answer? Obviously this one, serine. Correct na? Chloroplast, peroxisome, mitochondria. From the glycine produced here, glycine converted into serine here. Now friends, serine from the mitochondria, it is going back to the peroxisome. Very good. Okay, I only given the answer fast. Okay, sorry. Question number 39. Think and answer. It's a big question. Slowly think and answer. I think people are getting sleep. Some people left already. It's a simple question, friends. It's a simple question. if you come to know one option is wrong, you can easily come to conclusion of this question. Friends, you can easily come to the conclusion to this question. Okay, other girls, don't sleep please. Okay, anyone else can give the answer? 39. Friends, easy, friends, easy. Okay, you only tell me Rubisco is only one enzyme, right? And you just think it commonly with a common sense. How does one enzyme localize some part in one organelle? How does the another part of the enzyme is localized in the another area? How is that possible? It is not at all possible. Rubisco is present only at one area. You know, right, where the Rubisco is present, friends? In the stroma. Because we all people commonly know that stroma is the region where the dark reactions are taking place. Why do we require the nucleus first of all? Wrong statement. If you come to know, no dear, it's easy. If you come to know option B is wrong option, then you can eliminate option A also, option B also, option C also. Easily you can eliminate at least if you understand this option B. Why does the enzyme Rubisco has to be present in the nucleus? What is the role in the nucleus? Rubisco function is what? Fixing the carbon into glucose. Where does the carbon fixation will occur? Obviously in the stroma. Why in the stroma? All enzymes present over there only. Simple point. You can eliminate the option B very, very easily. Option A, you can come to a conclusion here, option D, but let me justify other statements. During the oxygenate activity of the Rubisco, I have very clearly told you, RUBP, how many carbon? 5 carbon. Whenever it is combined with the oxygen, what it will give? I told you, it will give one molecule of 3-PZA and another molecule of what? 2-phosphoglycolate. 2-phosphoglycolate is how many carbon containing compound? 2 carbon containing compound. We discussed very clearly. Option A is absolutely right statement, no doubt about it. I have explained the same line. Option C, the bound sugar phosphates in Rubisco are specifically removed by an ATP dependent enzyme, Rubisco activase. I feel you people did not know about this, but you remember Rubisco activase is an enzyme where listen my statements carefully. Rubisco activase is an enzyme where whatever the extra sugars that are added to the Rubisco, that extra sugar phosphates can be removed by the Rubisco activase enzyme. C is right, friends. Option D, the active form of Rubisco catalyzes carboxylation also, oxygenation also. Even though you feel option C is a difficult option, even though you don't know the option C, by eliminating the option B, you can directly come to a conclusion. Option D will be the correct and most appropriate. Okay, that's good. I hope everyone understand that correctly. Next, question number 41. Let me know if anyone did not understand. I will re-explain. Option D. Sadhguri, we have discussed, no dear. Rubisco function is what? Rubisco can fix the see, I will tell you one common definition. Sadhguri, the same line also I have told you. What is the full form of Rubisco? Tell me. The full form of rubisco is ribulose 1,5 bisphosphate carboxylase and oxygenase. This is the full form of the rubisco, right now dear, which means rubisco having the ability to fix the carbon also, rubisco having the ability to fix the oxygen also. When the rubisco fixes the carbon, there is no problem. When the rubisco fixes oxygen, then only the problem, right? Then we can call the photorespiration. That is what we discussed in first two classes of the photosynthesis. Now, when RUBP is how many carbon? 5 carbon. RUBP fixes oxygen, no carbon dioxide is available. Then rubisco, what it will do? This rubisco enzyme will take the oxygen. RUBP is how many carbon? 5 carbon. Then what we will get? We will get 1 molecule of 3-PZA and we will get 1 molecule of 2-phosphoglycolate. Simple. Hence what will be the answer? Sorry. Then what we can say, it can fix the carbon also, it can fix the oxygen also. It depending on availability. Same concept I have explained here. This is the enzyme. Enzyme having only one active site which is more available, that will come and fix associated with the active site, then rubisco will fix it. More the carbon dioxide will associate. More the oxygen associate. If you feel confused, please watch the recorded classes of the photosynthesis. You will get a clear cut idea. Okay. Go for question number 41. I think Shafia has given. Shafia, your answer is for what? 41, na? Where did I said 5 steps, dear? Where is the 5 steps? First of all, tell me. The active form of rubisco catalyzes carboxylase or oxygenation reaction in 5 steps. Okay, this one you are talking about. See, 5 steps in the sense, look into this. Once we get 1 3-PZA, and what we will get? We will get another 2-phosphoglycolate. Now the question saying that this 2-phosphoglycolate can be converted into 3-PZA in 5 major steps in the 3 organelles. That is, they are saying in 5 steps. You got my point? Okay, I did not see this. The 2-PZA converted back into 3-PZA in the 3 organelles by 5 major steps. That is the point. You got that? Okay, that's good. Question number 41. See friends, a Casparian strip obviously block what pathway? Apoplastic pathway only. There is no doubt about it. Of course true. Symplastic pathway means plasmodesmator. That's good. Okay. Question number 42. Okay. Tell me Sadhguru, if you don't understand, I will re-explain. Okay, that's good. Question number 42. Come on friends, please be active for some time. What will be the answer for question number 42? Sorry, what will be the answer for 42? It's absolutely true, sugars. Which one of the following component is expected to be the most abundant in the phloem sap of a plant? Which is the sugars. Amazing. Okay friends, question number 43. It's a kind of a very logical question friends. I want you to think and answer this. Option A only ABC. I will give the option. option B, only B, C and E. Then option C, only A, B and D. Then option D, only A, C, only A, C and E. Think it, friends, think it and answer. I don't know why I will press this, so sorry. Think and answer. Everyone given the answer for question number 43. Oh, super. Sumaya, your answer for 43, dear? B. Think it, think it. Okay. Okay, that's good answer. Look into this question number. Mrinalini, what is the answer? Others, Deepika. Okay, good. Following observations were recorded while studying physiological parameters of sorghum and wheat under similar conditions. And what's the given question? Given options? If you look into this, sorghum rubisco exhibit relatively higher affinity for carbon dioxide compared to the wheat. Right statement only, na? See, actually, friends, sorghum is what plants? Sorghum is C4 plant, correct, na? And a wheat, it is what? One second. Yeah, right. Sorghum rubisco exhibit relatively higher affinity for carbon dioxide compared to that of wheat. See, friends, sorghum is what kind of a plant? Sorghum, millet, sugar cane, these are all C4 plants. When it comes to the wheat is what plants, friends? C3 plant. But they said that relatively higher affinity. No, right? No, it's a wrong statement. Then option B, light saturation of net photosynthesis flux is relatively lower for the sorghum compared to that of the wheat. Of course, true. What is the reason? You all people know very well. In the C4 pathway, there is a no photorespiration, which means in the C4 pathway, there will be a no oxygen fixation. But in the C3 pathway, in the C3 plant in the wheat, there is a chance that photorespiration occur. So that's why light saturation of net photosynthesis flux is relatively lower for the sorghum compared to the wheat, because in the wheat photorespiration is possible. And option B is right. Warburg effect is difficult to record for sorghum and could be said as not measurable, where it could be easily recorded for wheat. First of all friends, kindly don't get confused with this particular Warburg effect. Warburg effect is basically used to understand the oxygen effect only. Correct na? Now you all people know that in the C4 plant sorghum, there is a no fixation of oxygen. If there is a no fixation of oxygen, then how can we study the oxygen effect? Not possible. But in the C3 plant, oxygen fixation is possible. Then we can study. B is right, C is right. Temperature optimum for net photosynthesis is lower for sorghum compared to that wheat. No, temperature is not at all optimum for the sorghum. It's higher for the sorghum. Option D is wrong. 13 carbon 12 carbon ratio of assimilate is relatively higher for sorghum compared to wheat. So friends, we know, right, very common point. C4 plants will efficiently fix the carbon dioxide, no doubt about it. C3 plants are not efficient in fixing the carbon because they will show the mechanism of photorespiration. Then obviously this statement is right. C4 plants have high carbon ratio, 13 carbon and 12 carbon, whereas in case of C3 plants it is low. Hence option B, option C, option E, three statements are correct. Hence the answer B is right. That's good, that's good, that's good. Why I am unable to see the link? Just give me a moment. Okay, Sumaya, there is a question is coming here about the compensation. I will explain here. Okay, see Sumaya, there is a question you can see here. Here I will explain. Okay, compensation in this graph I will explain. Okay, don't worry. Let's go down there. I will explain. There are a lot more questions are there. Please be patient, friends. Please be patient. Okay, so what we were discussing and where we are. Oh, this much fast I came. Okay, perfect. Next, question number 44. This is also kind of a logical question, friends. Think. C statement. 1 minute, Jayashri. C statement, Jayashri, listen dear. Warburg effect is basically used to study the oxygen effect in the plant body, or it is basically study the oxygen fixation. But you know, right, in the C4 plant we don't show any kind of the photorespiration, which means that we don't show any kind of a oxygen fixation. If there is a no oxygen fixation, then how can we show the Warburg effect? Warburg effect is difficult to record in the sorghum because there is a no oxygen fixation. But it can be easily recorded in the wheat because wheat is the C3 plant, photorespiration occur. You got it, dear? Okay. You know, friends, this respiratory quotient you people read in the 11th standard. In the 11th standard, you all people read about the respiratory quotient. First, for all of you friends, I want every student first listen my words. First just listen to me, then you people can answer my questions. First, what is respiratory coefficient? All the student has to listen. How much volume of carbon dioxide get eliminated? Please remember, how much amount of the volume of carbon dioxide get eliminated by how much amount of volume of oxygen is getting consumed. This is what exactly we can call the respiratory coefficient. We studied many times, friends. I will just write the values for the carbohydrates and each and everything. Respiratory coefficients for the carbohydrates. How much it, friends? It is approximately 1. Whereas respiratory coefficient for the fats, it is approximately 0.7. And what is the respiratory coefficient for the protein? It is a 0.8. Okay, so remember these three and try to answer the question now. It's a logical statement, friends. You need a better understanding of this question, but try it. Try it, it's okay. Answer B. Okay. Can anyone explain? Okay, anyone else? Very good, very good. The answer will be B. Fantastic. See, respiratory coefficient for the partial carbohydrates breakdown, it cannot be infinity. So you can eliminate the option A also, you can eliminate the option D also, just like that. Respiratory coefficient indirectly provide the information about what is the nature of the substrate, the relative rate of competing respiratory process. B is right. Yeah, it's more than 1 for the organic. That's absolutely right. Right. Then breakdown of organic acid in mature fruit will exhibit a respiratory coefficient value more than 1, since organic acids are relatively oxygen rich. Statement C is right. Anabolic metabolism never influence the respiratory coefficient. It always, we can say, decrease the respiratory coefficient. Hence option B is correct. Option C is also correct. Hence the right answer is B. Question number 45. Think and answer this question. Yes, dear, it is infinity is wrong, dear. It cannot be infinity all the time. That makes sense. I understand what is your question, Brunalini, but it cannot be infinity all the time, dear. It cannot be infinity all the time. Okay, question number 45. Oh my God. Okay, just give me a moment, please. Just a moment, friends. I just missed something. Okay, I got it. Question number 56. What will be the answer for question number 45? Amazing. Okay, question number 45. The answer will be B. It is not D, friends. It's not D. Angiosperm contains sieve plates. and there are no sieve plates in the gymnosperms. I already told you, friends, sieve plates are abundantly present in the higher developed organism. So what are higher developed organism when compared to the gymnosperms? The higher developed organisms are undoubtedly angiosperms only. So angiosperm contains sieve plate pores. So since angiosperms not contain the sieve plate pores are not present in the gymnosperm. This is right statement only, friends. Why the statement is wrong? P protein, I have told you, friends, P protein, it is basically prevent. I want every one of you remember, friends, P protein prevent puncture or damage. It prevents puncture or damage of the sieve tube elements. It will prevent the puncture of sieve tube elements. So P proteins are not present in the higher organism. Why? What is the reason? Because, friends, angiosperms are already highly modified, very highly modified structures. Since they are highly modified, you all people know, right? They are having a highly developed vascular system, which we read in our 11th standard also. Angiosperms are always having highly developed vascular system. In the gymnosperms, there is a poorly developed vascular system. So wherever the vascular system is poorly developed, there is a chance that phloem can rupture, phloem can damage, then food can be leaked out. So that's the reason what do we require? P protein is required in the all eudicots and many monocots. That's good. Hence the correct answer will be B. Amazing. Great. Next. Okay, this question is actually, this is not the plant physiology question. I kept it here. Leave that 46 question. Go for 47. This is not there. 47. It's easy question, friends. Just understand. It's a common question which we read in our 12th standards. Silicon. But we always tend to neglect this. See, friends, I want every one of you has to remember one common point. Whenever there is a no silicon is present, and friends, all the students listen. First of all, where the silicon is present, it is deposited in the ER, it is deposited in the cell walls also. Friends, if there is a no deposition of the silicon, plant will undergo a process called as lodging. What do you mean by the word process called lodging? Lodging means, friends, fall off. What, friends? Lodging means what do we can call it? Fall off process. So when there is a silicon, obviously it contributes the cell wall to become rigidity and elasticity because it is deposited in the cell wall. So as a result, if the cell wall is stronger, plant never be weaker. If the cell wall if it is not rigidified, cell wall become weaker, obviously the entire plant cell become weaker. There is a chance that the plant undergo the process of lodging. Question number 48. Think and answer. Think. Friends, even though you don't know this question, try it please. Try this. See, leucine rich repeats. Okay. Anyone else can give the answer for this? Okay, the answer for this is the transcription repression domains. It's not a RO gene product, it's just R. Most of the plant disease resistance gene product contains, they contain transcription repression domains. See, actually friends, the leucine zipper domains are basically act like a active domains. Active domains. Because friends, every student listen my statements carefully. See, everyone listen. I am a plant body. Inside my body there is a particular nematode got attacked me. Now I have to inhibit the growth of nematode, correct or not? How can I inhibit the growth of nematode, friends? I can inhibit the growth of the nematode by blocking its transcription. Very simple. So friends, the most of the disease resistant plants, they contain transcription repressor domains which can block the transcription of that particular nematodes or whatever that is attacking the plant body. Leucine rich domains are there actually, friends, active domains, so they cannot play any role. I want all the students remember transcription repression domain. Generally remember, I am a plant, I was attacked by a nematode, I was attacked by an insect. Insect release its toxin, I want to block that. So for that, I have to block that particular process by repression only. Hence the answer will be B. Don't confuse. Just remember. Question number 49. They are asking which one is not bioactive, friends. Not. Which one is not bioactive? We discussed many kind of gibberellic acids are always in the inactive form. Otherwise, they are acting as a precursors for the active forms. Fantastic. So friends, I hope you few people quickly recall, I have told to all of you that there are more than 150 types of gibberellic acid derivatives are there. More than 150 type of gibberellic acid derivatives are there, and I have told you to all of you friends, GA3, gibberellic acid 3 is the most widely studied. Most widely studied is the gibberellic acid 3. Most active form is what? Most active form. Most active form is the GA1. Okay, along with the GA1, GA3, even GA4 also biologically active form only, which is not biologically active form, which is inactive or acting as a precursor, that is the GA5. That's good. Question number 50. Come on friends, please be patient for some more time. We know this and I told you already. They are asking which is not involved. It's a good question. It's a really good question. Question number 50. Okay, that's good. I feel every one of you remember my statements. When we studied the structure of the nitrogenase enzyme, what we have studied? We studied that nitrogenase enzyme contain two centers. One is iron sulfur center and another one is the molybdenum iron center. This is what I told to all of you. I hope you all people remember this. Then molybdenum is right, iron is right. Friends, vanadium. I am using a word nitrogenase, right? There is an enzyme called, friends, vanadium nitrogenase. I want you to remember this. There is an enzyme called as vanadium nitrogenase, which we can use in the industrially to convert. That's what, dear. No. Brunalini, that's what I am saying. Vanadium nitrogenase is the enzyme which is involved in the conversion of N2 to NH3 in the industrially. There is a specific enzyme is there. We called vanadium nitrogenase enzyme. So here the answer will be cobalt. We don't use the cobalt. Very, very simple point. Okay. Then the question number 51 says that which one of the following is a fungal disease of the plant? Which is a fungal disease? What's the answer? Oh, everyone given the answer for this question number 51. Good. So apple scab is the fungal disease which is basically, it's a very direct question, nothing to explain over there. It is basically caused by the Venturia inequalis. Very good. Remaining are bacterial and viral diseases. That's good, that's good, that's good. Okay. Question number 54. Be patient, friends, please. You can easily eliminate this. Okay, anyone? What is the answer for 66? Sorry, 54. Answer for 54. 54 is D. Okay. Absolutely right. Friends, we can easily eliminate the option. What is the ultimate goal of the PPP pathway? Ultimate goal of the PPP pathway only production of NADPH, no? That is their ultimate goal. Hence the option B is wrong. If you comes to know that the option B is wrong, we can eliminate the three options just like that. Hence the answer will be A. Metabolism of glucose into pyruvate through the glycolysis generates, we know, right, two molecules of NADPH. A is right, B is wrong. Cyanide form a complex with heme iron of cytochrome oxidase. Absolutely right. Alternative oxidase is insensitive to a cyanide and has higher potassium than that of the cytochrome oxidase. Right statement. Hence the correct answer will be D. Okay. This is a very old question, friends, but see, this is about the photomorphogenesis and this question was not included in the syllabus. Actually, this is before 2011. So it was not there. No need to worry about this. Question number 56, because I kept some of very previous questions also that was not there. It's a good question, friends. Think it. Think it slowly, friends. Think it slowly. See, friends, we read now. I have told to all of you what is the function of a chlorophyll. The function of a chlorophyll, upon absorbing the light energy, chlorophyll is going to convert the light energy into chemical energy that can be used by the plant body, and they produce the ATP. Later they will fix the carbon into glucose. We all people commonly know it. Now just listen to me, just listen. And we all people also know very well, I have told you very clearly how much amount of the light energy can be absorbed by the chlorophyll, how much amount of the energy they can convert into chemical energy. Only that much it can absorb. It never absorb the extra amount, which is absolutely a true point. In the photosynthesis, I think I have explained it in the yesterday class also, in the sensory photobiology. When three chlorophyll molecules, when more chlorophyll molecules are exciting more amount of the energy, they get excited. See, one chlorophyll molecule got excited, second chlorophyll molecule also get excited, third chlorophyll molecules also excited. We discussed in the photosynthesis. Now these three molecules together, what do you call it as? We call it as a triplet. What is the function of a triplet? Triplet is the one which is going to activate free radicals. What it is going to activate? It is going to activate the free radicals. Never ever forget the point. What the free radicals will do? They will inhibit the entire photosynthetic machinery. So when compared to the single chlorophyll, triplet having the greater lifespan, always remember. So single state is short lived compared to the triple state. This statement is a right statement. Single state is long lived, wrong statement. Single state contain electrons. This is the only point I want you to remember. Single state contain electrons with an anti-parallel spin, triplet contain parallel spin. This is a direct characteristic feature. There is no need to explain over there. Always more chlorophyll molecules, they will spin always parallel. Less chlorophyll or a single chlorophyll, spin the electron always anti-parallel. Hence friends, C is the right answer. Option A, option C is the correct. Hence the correct answer will be C. Got it? Simple question. Now you people remember that without fail. Good. Just pay some more attention. We will fill some more questions and I will stop it. Question number 57. Question number 57. If you understand one option in this question, you can easily eliminate it. If you understand one option also, you can easily eliminate it. They are asking correct, friends. They are asking correct statements. Asking correct. Try to eliminate one option correctly, friends. We read this, friends. More than 3 options we read. Think little bit, think little bit. Of course, that's good. Perfect, perfect. Option D. Friends, we discussed, right? Where is that? The metal associated with enzyme nitrate reductase is not at all magnesium. It requires actually molybdenum. So option C will be absolutely a wrong statement. Hence, you can eliminate option A, you can eliminate option B. Now look into the option A. Nitrogen is normally taken by the plant in their fully oxidized form, but needs to be reduced before incorporation into inorganic form. What is that reduced form? Reduced form only NH3, NH4, NO2 and NO3. These are all what? Reduced forms. When we reduce the nitrogen, then only we will take, otherwise we can't take. Option A is right. Option B, conversion of oxidized nitrogen into reduced nitrogen needs energy in the form of NADP plus. No, right? We will take the energy in the form of ATP. So option B is wrong. Option C is wrong. Nitrate reduction takes place in the cytoplasm. Why? You all people know why the nitrate reduction takes place in the cytoplasm. The reason, listen my statement, nitrate reductase enzyme is present in the cytoplasm, whereas nitrite reduction takes place where? In the chloroplast. Why? Nitrite reductase enzyme present here. Simple point. Hence the D is also correct. Option A is correct and D is correct. This is the answer. Okay. Okay, friends, is that clear everyone? I think it's almost 3 hours. Okay. Is that clear everyone till this question? There are very little number of questions are there, friends. We will solve these questions in the tomorrow class. Okay, I hope it's clear for everyone whatever we discussed so far till this question number. I want you people to remember the question number 57. Okay. Okay, friends, that's all for now. I really appreciate everyone patience, so which we require during our exam preparation. Okay, so I really appreciate it. We will meet in the tomorrow 8:30 a.m. in the Unacademy class in the human physiology. Okay, I will see you at 8:30 a.m. sharp. Is that clear? Okay, friends. Okay. I hope the session is little useful to all of you. Okay. Okay, friends. Thank you all. Everyone sleep well. Okay. I will see you in the tomorrow 8:30 a.m. Bye bye. Thank you all. We'll do that, dear. Surely I'll do it. Okay. Bye bye. Thank you so much. I really appreciate every one of your patience.